Students, today we are going to start our next chapter of plus 1 class biology, and the name of the chapter is plant growth and development. In this chapter we will mainly cover about the main topic, that is plant growth regulator, or we can say plant hormone or phytohormones. This topic is already discussed in previous classes also. In 10th class there was a topic phytohormones or plant hormones in your biology book. So this topic is related with that 10th class topic. So let's start our first topic, that is plant growth regulator. As the name suggests, what are plant growth regulator? These are some chemicals which help in regulating the growth in plants. So plant growth regulator are the chemical substance that influence the growth and differentiation of plant cell, tissue, organ. Plant growth regulator function as chemical messenger for intercellular communication, means they help or they work as a communication mode, or you can say these are the some chemicals which help in transferring the message from one part of the plant area to the other part. Second, next point, they work to coordinate the growth and development of cell. It means plant growth regulator work in close coordination, means one plant growth regulator will help the other plant growth regulator for proper growth and development in cell or in plants. So plant growth regulator are of different type or different derivative. They may be indole compound, that is indole 3-acetic acid. They may be adenine derivative, that is cytokinin. They may be derivative of carotenoid, that is abscisic acid, or terpenes, gibberellic acid, or some plant growth regulator are gaseous in nature, that is ethylene. So phytohormones or plant hormones are of 5 type. You can see in the slide: auxins, gibberellins, cytokinins, abscisic acid, and ethylene. These are the 5 plant hormones which we have to discuss in this chapter, and these hormones are very important. So we can categorize the plant growth regulator into two major group. One is growth promoter and other is growth inhibitor. Growth promoter means it lead to that activities which help in promoting cell growth, cell differentiation, cell division, or you can say elongation of cell or increase in the length of plant. So growth promoters are 3 hormones, that is auxin, gibberellin, and cytokinin, whereas growth inhibitors are of 2 type, abscisic acid and ethylene. Growth inhibitor hormones are those hormones which inhibit the growth of plant, or you can say inhibit the cell division. So there are 2 hormones which show growth inhibiting activity, and there are 3 hormones which show growth promoting activities. First is auxin. Auxin were discovered by Darwin and his son Francis Darwin, and auxin was first isolated by F.W. Wentz in the oat seedling. Further, Went also performed a curvature test for studying the activity of auxin. So in the lower part we can see these are some example of synthetic auxin, that is IAA, IBA, NAA, 2,4-D. 2,4-D is 2,4-dichlorophenoxyacetic acid. Now these are the functions of auxin. Auxin promote apical dominance. What is apical dominance? It means it help in the growth of only the tip portion of plant. It initiate rooting in stem cutting. It also promote flowering, for example in pineapple. It also prevent fruit drop and leaf drop at early state. It also promote abscission of old mature leaf. It also induce parthenocarpy. Parthenocarpy is the formation of fruit without seeds, your seedless fruit production. It is used as herbicide, means auxin as a chemical can be used for killing the unwanted plant. It controls xylem differentiation and help in cell division. Next, gibberellin. Gibberellin was discovered by a scientist named as Kurosawa, a Japanese scientist, and gibberellin was extracted from a fungus named as Gibberella fujikuroi. So, according to the name of fungus, the chemical produced by this fungus named as gibberellin. Gibberellin was isolated from infected rice seedling. Now, what are the physical or physiological functioning of gibberellin? Gibberellin help in elongation of dwarf stem, means if a plant is short in height, then we can treat this plant with gibberellin so that the plant will elongate. We can overcome the natural dormancy. What is dormancy? Dormancy is the state when plant unable to grow if we provide favorable condition also, means if we provide favorable condition to the plant but plant will not germinate or plant will not grow, this is known as dormancy. So it overcome the natural dormancy of bud, tuber and seed. It help in seed germination. It induce parthenocarpy, means development of seedless fruit. It promote flowering in long day plant, and it help in promoting the vernalization. Vernalization will be discussed at the end of this hormone topic. Now, cytokinin. Cytokinin was discovered by Skug and Miller, and cytokinin, as the name suggests, it show the activity of just like a cell division activity, and cytokinin activity was discovered in coconut milk. Coconut milk help in promoting the cell division. Now, physiological effect of cytokinins are as follow. It induce cell division. It delay the senescence of leaf, means aging. Senescence means aging of leaf and other organ. It promote lateral bud. It increase cell expansion in dicot, and it promote the chloroplast development and chlorophyll synthesis. Now, ethylene. Ethylene is a volatile plant hormone, means it exists in gaseous state, and it help in inducing the ripening of fruit. So now, what are the physiological effect of cytokinin? It promotes senescence, means aging. It inhibits the elongation of stem and root. It induces fruit ripening, means if unripe fruit is treated with ethylene gaseous hormone, then the unripe fruit will convert it into ripened one. It breaks seed and bud dormancy. It initiates germination in peanut seed. It helps in sprouting of potato tuber. It promotes root growth and root hair formation. It initiates flowering and synchronizing fruit set in pineapples. Next is abscisic acid. Abscisic acid was discovered by Frederick T. Endicott and his co-worker. Abscisic acid is a growth inhibiting hormone, and when the plant is in stress condition, this hormone will release in large amount so that plant can cope up with stress condition. Now, functioning of cytokinin, it act as general plant growth inhibitor. It induce seed and bud dormancy. It inhibits seed germination. It stimulates the closure of stomata. It play an important role in seed development, maturation and dormancy. Next is photoperiodism. periodism, we can split this into two words. Photo means light, periodism means duration. So different plant respond different according to the relative length of light and dark period. So photoperiodism is the response of plant to relative length of light and dark period. Means some plant will show flowering in longer dark period, or some plant show flowering in longer light period. So plant require a definite period of exposure of light to induce flowering. So photoperiodism is on the basis of light duration. Plant is of three type: long day plant, short day plant, and day neutral plant. Long day plant, as the name suggests, it require exposure of light for a period exceeding the critical duration. Short day plant, it require exposure of light for a period less than the critical period. And day neutral, these are the plants which have no relation between day and night time. Now vernalization. Vernalization is the phenomena of inducing the flowering in plant by exposure to low temperature. Means some plant require low temperature for inducing the flowering. So we can provide the low temperature condition according to our we can say requirement. So example are biennial plants. Biennial plants can be induce flowering by exposure to low temperature. These are the monocarpic plant that flower and then die in the second season. Example: sugar beet, cabbage, carrot. So this is all about in today's lecture.